Hello, everyone. A very good afternoon to those who are joining us from Singapore and Asia, and good morning or good evening to all of you joining us from other parts of the world. Thank you for being here with us for today's online event on bioprinting and organic solution, presented by SGInnovate and Catalyst and partnered with the Netherlands Embassy in Singapore. And we have Barbara from the Embassy here, and she'll have her contact information in the chat box for you to reach out to her. My name is Jin from SGInnovate, and as a Singapore government-owned organization that invests in and helps build deep tech startups, talent, and community, we have been building up and driving deep tech innovations in AI, healthcare, quantum tech, agri-food, autonomous technologies, just to name a few. At SGInnovate, much of our work is to connect Singapore with the global deep tech ecosystem to learn how we can better harness technology. Today, we have our panel of experts from Singapore and the Netherlands to shed light on the potential for bioprinting to become the latest groundbreaking solution, how it is set to revolutionize the healthcare industry, and the opportunities that arise out of this up and coming market. Throughout the session, we encourage you to engage with our speakers by submitting your questions in the Q&A box located on the lower panel of your screen, and we will only be taking the questions from the audience at the end of the panel discussion after the presentations. But feel free to submit your questions through there anytime throughout the presentations. So without further ado, I would first like to invite Azra from Catalyst to give a quick introduction. Azra, please.
Thank you, Jin, and welcome everyone to today's session. I'm really excited for this session because we do have Biotech Connection Singapore, SGInnovate, and also the Netherlands Embassy in Singapore, which are also one of our partners here at Catalyst. Let me just give a brief introduction of what Catalyst do. We are a first of its kind innovation hub for medtech and health tech ecosystem, and it's a collaboration between Padang & Co and Aardvark Labs. And we aim to bring together clinicians, innovators, and ecosystem partners, just like SGInnovate, to create new partnerships as well. So I will not take up too much time as well, and please enjoy the session wherever you are. And yeah, Jean, back to you.
Thank you, Azra. And we'll start off with our very first presentation by Dr. Ricardo Lovato. So over to you, Ricardo.
Thank you, Jean, and good afternoon to everybody. My name is Ricardo Lovato. I'm from the University Medical Center, Utrecht University, where I'm associate professor, and I'll be happy to share some of our work with you. Just a second. You should be able to see my screen by now in the presentation mode.
Yes, we can.
Perfect. So thank you again for this invitation, Jean and Barbara, and thank you all for being here. So what I would like to discuss with you very briefly is a bit what we do in Utrecht in the field of bioprinting, and give a bit of an insight on how this technology can help healthcare as well. 3D printing is already a reality in medicine, and probably all the attendees here know it. That is nowadays possible to envision the 3D printing of patient-specific prostheses, but also doctors can start from medical images to recreate 3D models, and they can touch and feel with their hands before actually going to the surgical theater. So they can actually train looking at the anatomy of the site that they want to operate, not just from a virtual image, but from an actual object. What we try to do in bioprinting, though, is to merge this world with the world of tissue engineering. Tissue engineering, you typically have this paradigm which you can take cells from a patient, like stem cells, expand them in vitro, combine them with proper 3D materials that mimic the metrics of tissues, and then culture them to mature and reimplant them to fill, for example, a tissue defect or a disease organ. Now, what we do in bioprinting, instead of using metals, polymers, and ceramics only as classical engineering materials, we actually make these cells and biomaterials into our printable inks, so something that we use as building blocks to create 3D objects. Now, if you do that properly, and if you combine multiple cell types, you can actually recreate something that very closely mimic the composition at the cellular level and at the geometrical level of actual tissues. One thing that is important to remember, though, is that unlike what happens with conventional 3D printing, the object that comes out of the printer is not ready to be used yet. It's not an actual organ. It may look like such, but it doesn't behave as that. So the main challenge right now, which is still the biggest challenge right now, in my view, is that we have fantastic tools and technologies that are exceedingly developing very fast to recreate with extreme precision and accuracy the composition of tissues. But we still need to use these preformed geometries to guide the maturation of these cells to eventually create something functional. So in this cartoon, for example, a muscle able to contract. And 3D printing can help us because we can select multiple cell types, and different cell interactions are fundamental in maturation towards tissue organogenesis. We can develop different technologies that allow us to target features at different length scales, from the micron scale to the macro scale of living tissues. And importantly, we can combine multiple materials to create, for example, interfaces between, for example, bone and cartilage. So the tissues are not sitting in the void, they're connected to neighboring tissues and organs. We can provide channels for nutrient supply and vascularization, which is needed, of course, to provide survival of larger constructs. And we can provide mechanical support for those tissues that need also to perform a mechanical function, such is the case of the musculoskeletal arena. And of course, we can aim to scale up this tissue. We'll briefly touch with some of the research that we do to give some examples on these different fields. In Utrecht, we have a biofabrication center where most of the technologies available nowadays are clustered, and we also develop new devices and materials that can allow for 3D printing, primarily in the field of bone and cartilage restoration, but we also work with different type of, oh, sorry, that didn't work out. I will share it again and go to the slide afterwards. But I was also saying that some of these tissues are also soft tissues, such as liver, for example, that I will show you in a couple of slides as well. So the simplest thing we can do is actually to start printing inorganic or organic scaffolds where cells are seeded afterwards. And that's not really bioprinting, but it's still in the realm of biofabrication. So you 3D print, for example, in this case, a ceramic material that is biodegradable and mimic the composition of bone. We can culture cells on top of it and make them differentiate towards bone cells, and implant them in even in large animals, in this case in a critical size defect in a horse bone. That's about 1 centimeter scale in both in diameter and in height. We can implant them and eventually observe how new tissue it regenerates over time, and the porosity of the scaffold is embedded by new bone as the material degrades, and is digested by these multinucleated cells that you see here, which are actually macrophage osteoclasts that are actually degrading the ceramic component. This is more or less the most standard way to do 3D printing in combination with tissue engineering. But we can also think of technologies that actually include cells in the printing material. So in that case, for example, in this example, we work with gelatin-based hydrogels in which suspend liver organoids. So liver organoids are, and in general, organoids are microscale aggregates of cells that are not just spheroids, but are actually able to reassemble themselves and create multiple cell types that mimic the function of a certain organ. In the case of liver, we start from hepatic stem cells from adult patients or healthy people, depending if you want to model a disease or a healthy liver. And these grow into these spheroids that more or less look like what you see in this picture. So these cell aggregates, they're cystic structures, so they're actually hollow inside. And they have a polarization so that one of the sides is the one that deputed for the uptake of different nutrients or toxins or anything else that the liver compound needs to metabolize. Importantly, these aggregates, we work together with the group of the veterinary faculty in Utrecht, the group of Bart Spee with this technology, are actually able to express a variety of important mature hepatic markers. Here, just an example of one of the cytochromes that is deputed in the metabolism of drugs, and they're able to respond to different type of drugs. So this is a very simple test done in the liver field. So we give acetaminophen, the main component of paracetamol, to these organoids that have been printed in these porous structures, and they're able to convert acetaminophen to its first metabolite in the liver, which is also toxic for the cell. So we can actually assess that there is a differentiation ongoing, and indeed the toxic compound is released in this case. This is just an example. So you can, for example, create these large platforms that you can use for drug screening. But we also work more on the further development of these technologies. So right now, 3D printing is mostly done either by extrusion or by digital light projection technologies, which is what you see here playing in these videos, which are extremely powerful, but they're also relatively small. So if you think about it, actually, 3D printing was born more or less in the 70s, between the 70s and the 80s of the past century. It doesn't really evolve that much since, even though now we, of course, can print cells and gels, which is, of course, a big advancement. But the technology still works in a layer-by-layer fashion. And when your materials are living cells, well, if you work with particularly slow technologies, and you want to create large constructs that can be used for clinical purpose, for example, like you would see in this case, we can observe that cells sitting in the cartridge of the extrusion printhead, if they stay there for too long outside of their optimal culture environment, they tend to lose some of their metabolic activity. So that's why we started working at faster ways to 3D print objects, and that's what we developed a technology termed volumetric bioprinting. We joined strength with a group in Lausanne in a PFL that was expert on photonics, and we brought in our experience in bioprinting, and we developed this technology that is based on tomography. What you see here in this video running is actually a real-time print of a tower from the city of Utrecht. And basically what we do is we project layer light through a digital micromirror device onto a rotating vial containing our gel with cells. The different projections that we get from the tomographic pattern make a sort of an hologram. It's not the right physical term, but it gives a bit the idea. And where this hologram appears, or this combination of projections is providing the cross-linking of the gel, and everything else can be washed out. So we have our object forming very rapidly. And this is a very rapid process. If you compare this auricle model, for example, up to 4 centimeter cubes, we can print it in less than 25 seconds. Whereas if you want to print the same object with extrusion or digital light processing, that can take any time between a few five minutes and up to even a couple of hours, depending on the size. And importantly, we can also print very smooth features. So we don't have the typical filament size or the pixel size that you see in the conventional printing methods. And we now reach resolutions in the range of 100 micron, more or less, which is comparable to extrusion-based printing. Importantly, we can put cells in that and preserve their viability. We made some proof of concept in the recent past, where we work with this technology to recreate this complex trabecular bone structure that is here reproduced with a micro CT scan. And inside what appears white in the micro CT, which is again is a gel based on gelatin, we have this 3D printed osteoblast derived from mesenchymal stem cells. And in the porous network, you can actually add vascular cells, endothelial cells that eventually create their capillaries that start penetrating into the gel and being attracted by the bone cells, start integrating in the tissue. And this is now taken a step forward in the ERC project that we just started this year in the lab. The same technology is also now being explored in a larger consortium. It's a European consortium funded by the European Innovation Council named ENLIGHT, in which we are looking at applications for soft tissues. In this case, we are looking at bioprinting of pancreatic organoids within this vascularized pancreas structures that you see here represented in this picture. And the idea again is to create an in vitro model that can be used to test new drugs for diabetes that are developed by our industrial partners, but also to provide the first proof of concept of a transplant of this pancreatic unit, where we want to see the ability to regulate glucose metabolism in a diabetic mice in this case. This is just to give you a bit of an overview, and I'm happy to go more in details during the panel session and the Q&A session with you if you have any question on all these technologies. And I would like to thank you for your attention and give back the floor to Jean.
Thank you, Ricardo, for your sharing. And to all of you tuning in, please feel free to type any questions that you may have in the Q&A box, and we will be taking those questions after the presentations. Next, I would like to invite Dr. Javier Fernandez from the Singapore University of Technology and Design to share about his work. Javier, please.
Okay, let me share the screen. Can you see my screen?
Yes, we can.
Okay, so hello everybody. I'm Javier G. Fernandez from the Singapore University of Technology and Design, and I'm going to try to give you like a different perspective of bioprinting, actually, like a little bit like out of what most of the people understand bioprinting is for. So if you think about like what is the perspective for bioprinting, you will think about something like this, right? It's usually associated to like printing an organ. So it's basically like called like the future of organ transplant, right? And is is what is even more interesting, right? Is open this door for making organs using the cells of the patient, right? So you potentially could reduce or like eliminate everything that is related to rejection of an organ implant, right? So, and the idea is that we are going to build those organs by additive manufacturing. We are going to use an additive approach, right? This idea actually is not new, right? So this work here is my work in like 11, 12 years ago, right? So, and the idea of like building organs block by block, it was already there, right? So bioprinting, as we know it these days, it was not even a thing. So, but the idea was there. So the point was very simple. So we are going to take cells, we are going to encapsulate, we are going to make blocks with these cells, right? And we are going to assemble these blocks to make an organ, right? We are going to make them like to follow certain geometries to like basically get the shape of the organ that we want. In order to do that, we use capillarity forces, because those capillarity forces are something that is very neglected in our day-by-day, but at the micro scale, they are extremely strong forces. So we can control the surface of our system, we can control the blocks or the cubes that we are doing, and we can actually make them assemble in the way that we want them to assemble, right? I call the technology micromasonry, but then with time, the community started to call it bio-Legos, because I don't know, why not? So so the thing is, like, it was successful. I mean, we were able to aggregate these cubes. We were able to make the geometries, right? We were able to combine different type of cells, and we make like heterogeneous systems, complex systems, and the cells were surviving and they were healthy. And it's kind of like a good result, right? But if you think about the final result, right? This is not enough to build a tissue. So, and that is something that we realized really early in this field, that putting the cells in the right place is only half of the story. That's not an organ, right? So, and this idea comes from what we have learned in the school. So when we go in the school, we learn about the human body, and we get this representation of the human body as an aggregation of organs, right? And then if one organ gets bad, you can basically remove it and then put another or replace the organ, right? And then like the organs are made of cells that they are like in a little bit like in the same approach, right? It's just basically a group of cells that they are organized in certain manner. But actually the human body doesn't look like this. The human body looks more like this, right? So this is just a map of part of the metabolism of the human body, right? So all of these lines, all of these molecules that are here, and probably like we are at the limits of the resolution that Zoom allows, right? But all of this is just part of the communication that is happening in your human body. And that's important because we are not the addition of separate pieces, right? We are a whole machine that is interconnected at multiple levels. And the organs are not an exception, right? So they are not different in that sense. So, and that's important to understand, because if we place the cells in the right place without those interconnections, without the interactions between the cells, we are not really building an organ. We are more like representing an organ. It's kind of like a painting of an organ rather than a functional one. That doesn't mean that we are not, or we will never be able to print an organ. Actually, like eventually we will be able. But the first question that we should be asking ourselves is if we are actually in the right path. Is our technology is going to be just a painting, or is going to be used to paint organs, or is actually going to be able to reproduce these interactions between the cells? So a couple of years ago, we embarked ourselves in a quest to actually answer this question. So we know that many of these processes here, right, are the result of cell-to-cell communication. So, and they are the result of mechanical forces. We know that cells can talk to each other, and they can actually drive the development based on that communication. And we know that mechanical forces, they have the same effect, right? So a perfect example for this is an egg, right? If you think about that like a chicken egg, right? So it's a perfect isolated system that starts with a single cell. And ends being a chicken, right? But during the whole process, we start with one cell that at some point becomes 2 identical cells, right? And then 100. And then at some point, the embryo decides that I need to start making some vascular system, right? And then I get like millions of cells. And then I start thinking maybe it's time to make like a nervous system, right? And all the information has always been in the genetic code of the cells, but the triggers that decide like make these cells behave in one way or the other are basically coming from outside, are coming from this communication. So last year we tried to answer the question, so can we reproduce these interactions with 3D printer, right? So what we did is like we took like one of the most precise 3D printers in the market, right, or in the world, and then we used it to produce artificial embryos. So, and we use the printer to basically give different inputs to the to these embryos, right? By constraining them to different geometries to basically control exactly how these embryos, they control exactly the environment of this embryo, right? And the idea was very simple. So if our tools are precise enough, we should be able to reproduce specific conditions on the embryo development and then trick the embryo to produce a specific type of cells, basically telling like, now you have like these conditions, now you need to do this, right? So in the ideal scenario, we should be able to find the conditions to produce a vascular system or reproduce the conditions to trigger the development of the nervous systems and everything related with the characteristics of the 3D printed surrounding, right? So the good news is that we actually like we managed to demonstrate that. So our target were cardiac cells, and we not only succeed in driving the whole development of the embryo towards the production of cardiac cells, right? But also those cardiac cells, they were extremely healthy, right? So you can see here, like, I don't know if it's clear in your screens, but this is a beating group of cells in our artificial embryos, right? So, and the beating is extremely constant and extremely large, actually. So, and that's amazing news, because it means that we have reached now like the point when with additive manufacturing, we can control the geometry or we can control an environment to a level that enable us to control the fate of a group of cells. And that's beautiful because they demonstrate two things. First, like the state of the technology, right? But also, since 3D printing is one of these fields that is growing like so fast, right? And we are being able to print faster, and we are able to like produce more more precision in our structures. It means that these communications is going to be getting better and better. So we are going to be able to drive this development more precisely, right? So I'm going to skip this because I think that I'm a little bit over time, and I want to like give time to the discussion. But this is like a small example of like the second part of 3D printing, right? That is the material. So let me go to a specific example that this is something that I want you to retain from this talk, right? So I just say that we know how we can work at a scale that is relevant to cells, right? But also at the beginning of the talk, right, I say that bioprinting is, the organs are really complex, right? And bioprinting is not there to reproduce that complexity. And that might sound like it's something bad, right? But it's not bad in the right context, right? So because our body is very complex. It's actually like too complex in many cases to fully understand many of the process, because there is a lot of noise everywhere. There are so many things happening at the same time that actually isolate a specific process is extremely difficult. So, but bioprinting is really good in doing that, right? Bioprinting allows us to make a replica of an environment in the body, and we just see in the sample of Ricardo these small samples of liver, right? That is basically a small area of the body that we can reproduce in very controlled conditions, right? And that's the key, because that allows us to remove factors and study or focus our study in the parts that we actually want to query, the ones that we want to actually understand, right? So to do this, and one example of this is what you have in the screen, right? So we basically reproduce the environment of a breast cancer. And we reproduce the environment of a cancer that is invasive and a cancer that is not invasive. And our goal was to, okay, if we are able to give like the same properties of the environment of the cancer, can we actually give some insights about like what is the difference between the cancer that is going to be metastatic, right? The one that is going to try to invade another organ, or the cancer that actually decide not to do that, right? What is the difference between these two cancers? Can we get that information by removing all the noise from the human body? So, and in order to do that, and that's another important part of the control environment, is that we when we build these mimics of the parts of the body, right? We don't need to be constrained by the geometries of the human body. In our case, we want to measure, we want to get a lot of data, and we are not going to do the analysis because humans don't do the analysis these days of all the data, right? So we want a system to be able to track those cells, to actually analyze those cells and get all the information for us. So we build a system that is a mimic of the human body, but at the same time is built for data acquisition. It's actually a system that is made to work in combination with machine learning, basically, right? To the data that we need for a machine learning process. So I'm already on my time. So, but basically the conclusion for that is that we can observe things that are completely impossible to see in vivo, or even in a system that is built for like human eyes, right? And one of the conclusions, for example, was that all these cells, even the invasive and the not invasive, right? All of them, they try to like find another place to migrate, right? But the ones that are successful, those cancers that we call metastatic, right? So are the ones that the cells are extremely focused on doing that. The ones that they don't produce metastases, they basically walk and they do it in a more random way, right? While the ones that they are successful in producing a metastasis, they actually walk in a straight direction always, right? They are extremely good in moving in one direction, even if they do it at the same speed and they walk the same distance than the other one, right? And I'm not going to bore you about like the algorithm and everything, but basically the conclusion here is that bioprinting has the chance, it's not only useful for put things inside the patient, but it's also extremely useful at this point to move medicine out of the patient, right? To know what drugs will work or not before using them, to know which doses do we need to use, when we need to administrate them, what kind of cancer is going to, what is a cancer is going to do, right? Before actually does it, right? So, and in order to do that, we need this perfectly controlled environment. So we basically want to build the patient outside the patient, and I think that's it. So I think that I'm already my 10 minutes.
Thank you, Javier. Thank you so much for that sharing. And as some of you in the chat box have actually pointed out, this is all an amazing combination of engineering and biology. And I don't want to take too much time. I will now pass over to Saujanya, who will be moderating the fireside chat to kickstart the conversation. Saujanya, please.
Thank you so much, Jen. First of all, I would like to really thank Dr. Ricardo and Dr. Javier for this really amazing and fascinating talk on bioprinting. I'm sure many of the audience here, many of them are there who don't know anything about bioprinting, and some of them who have an understanding, and some of them who are actually working on this topic. So I think one thing is for sure, this is a perfect game changer. And we do have a list of questions to ask you. But first of all, I think from your two talks, one thing is very clear, that the human body is super complicated, as how Dr. Javier actually showed. And that's one of the best pictures I've ever seen, actually, of how complicated our human body is. And also about stressing about how accurate and how the bioprinting actually can change the world for actually understanding the drug usages also. So before we start, actually, Dr. Ricardo, this question is for you. I was wondering if you could actually talk a little bit more about the Enlight project that you had mentioned, where you're moving from the in vitro to the in vivo proof of concept.
Yeah, so the project actually just started a couple of months ago. we have this pan-European consortium with 8 partners, 3 of which are industrial partners, 2 large enterprises and 1 SME, and the others are academic partners, where we are going to develop further this volumetric printing technology to basically pattern a vascularized network of engineered pancreatic islets that we develop from iPSCs, from induced pluripotent stem cells. So we generate these pseudo islets, we load them in our gels, and we print them inside the vascularized network that we can then use for perfusion to provide nutrients to these islets. What you're going to do with this engineered pancreatic unit is that the first and foremost thing is to actually use it for testing new drugs. So that would be in the direction of the organ-on-chip directions, more or less, that Javier actually very brilliantly explained before. So the idea is that you can now move bioprinting towards servicing the personalized medicine and provide models that can capture, of course, not the whole complexity of an organ, but some salient features and some key interaction between different components of the organ. For example, in this case, the islets and the vasculature, to see more in specific detail what the effect of these new drugs is in terms of toxicity, short and long term, but also functionality. And this is quite important because it can help us to either overcome the limitations of conventional 2D cultures, or right now, for example, in the field of pancreas, the golden standard is still isolated islets from human, which is outside of the body have an extremely short-lived lifespan. So that you will be able to perform only some acute toxicity assays, but you cannot test any chronic effect or long-term effect, and we can instead try to mimic it with our models. And that, of course, reduces also the need of using certain animal models, such as pigs in this case, that's used a lot for this field. The project is mainly focused indeed on this aspect, but then we also want to explore the direction of medical translation for regenerative medicine. So a version of this engineered pancreas will also be implanted in a diabetic mice, but that would be more towards the end of the project.
Absolutely. That's really a very exciting project, I should say, and it'll be very enlightening to understand how the results would go in this project. Thank you. So you do have another question coming from the audience, which is volumetric printing looks amazing, but how do you deal with vascularization if the gel substrate is homogeneous?
Mm-hmm. So the gel is homogeneous in terms of chemical composition, indeed. At least the ones I showed in this presentation. Vascularization right now we control it mostly to preform geometry. So we create vasculature patterns that afterwards we line with endothelial cells and supporting cells. That being said, we are also working towards a multi-material printing version of the volumetric printing. That's still work in progress though. That will indeed overcome this issue of homogeneity. So we'll be able to actually introduce anisotropy also from the chemical point of view or the biochemical point of view within the constructs. In that sense, keep in mind that bioprinting right now so 3D printing is 40 years old, more or less. Mm-hmm. Bioprinting has been around for 15, 20 years roughly, but has only become popular in the past 10 years. And the volumetric technology, it's only 2 years old. So there is still a lot of a very early stage development to be performed in this specific technique.
So the next question here is for both of you, for Dr. Ricardo and Dr. Javier. So which model make 3D printers are used for bioprinting? Are there any modifications that need to be made, and how many different materials can be printed in the same object or organ at this time? So this is open to both of you.
I can give my vision. I think that now is like the market is really populated with different makers, and the number of materials and the number of cells that you can put there is, I mean, I would say like the typical is one, but it's actually like finding like printers with 3, 4, 5 materials is pretty usual these days. I agree a lot with Ricardo. Like these days, the main issue is also the time that it takes to print. Like more materials means also more cells, means also more changings. So it's actually like longer to print, and you start like getting problems of your cells not surviving the process actually. In my experience, and maybe it's because from where I come from, most of the printers are built by the group. So I mean, obviously there are commercial ones, but still these days, most of the printers that I know in the labs, they are actually like built by the own lab. It's not hard actually. It's 3D printing is so well developed these days, right? So I think that yeah, that's my perspective.
Dr. Ricardo, would you add?
Yeah, if I can add something to that, indeed, yeah, as Javier said, that I think in terms of commercial printers, the one I can think of that has most printed at the moment is probably 6. So you can have 6 different materials at the same time. But if you make the printer yourself, that the limit is basically your decision. So if you want, you can make 20 printheads, and you just have to make a bigger printing area. And indeed, there is a lot of, if you're handy enough, you can build an extrusion printer yourself. It's relatively straightforward. There are also a lot of instructions through specific papers on journals like HardwareX, which they explain you protocol to assemble devices, for example. But there is also a very active community online that exchanges designs and prototypes. So if you have the people that have the skills in the lab, you will end up building the printers yourself, which is also something that we do as well in our lab.
Wow, that's really nice to know that you can do 6 at one, and then you can build this way. This is so. In the same line, I think there's another question that's come up saying, assuming certain organs are less challenging to bioprint than others, which are easier, and so they want to know which are easier and which are harder based on what your experience says, and what are some of the most difficult challenges you faced?
I'd say, if I can start, I would say none is really easy, in the sense that let's say it's as easy as you want to make it, in the sense that we can look at only one specific function of that organ, and then you can make a very relatively simplified model. For example, if you want to make maybe the example that is used of the simplest, it's probably the skin. The skin has a huge array of functionalities. Of course, if you just look at the barrier function, you can probably get away with a layer of keratinocytes and maybe the dermis underneath, and then it's a relatively straightforward thing to achieve. But if you want to look at all the actual functions of the skin, so sweating and temperature control and all the other functionality that are in there, and the vascularization and so on, then we are approaching that massive complexity that Javier exemplified in his talk. So yeah, it's easy. I would never use the word easy, but if you think of an example that is most advanced, let's say, that's probably indeed skin, for this reason, because at least the barrier function and the response to cosmetics or certain topic drugs can be tested, or at least has been demonstrated already that can be tested on these advanced models.
Mm-hmm. So it also depends, I think, on how many aspects we are looking at, as you just mentioned, I think.
Yeah, I totally agree. I think that is in general, like first thing, like tissues that are thin are easier. Like, for example, spleen was one of the first ones that it was produced, right? Because there's no cells, right, or almost no cells, and skin is also kind of like it's easy to control. And one thing that we have been discussing, and it has been like mentioned in both of our talks, right, is always vascularization. So the more 3D you become, there is like this block that has been in the field of tissue engineering for the last 20-30 years, that is angiogenesis. Is like, it's easier to make something in a lab, right, or like keep it alive, but then as soon as you start getting bigger and bigger, you start having problems of like moving oxygen, moving nutrients. You have like all these problems for building large tissues, right? So if you add so that is going to be, or that is definitely like one of the main challenges. And then if you add that, like the complexity that we have been talking during the whole talk, right? Like when we see an organ, right, it looks like a really homogeneous material, but then when you go in detail, you have like, it has like all this ultra structure that is in many cases is extremely difficult to reproduce, right? So yeah.
Yeah, I think it was quite clear from your talks, based on especially when you were staining them with E-Catherine and then DAPI. So it was very clear that you're showing the vascularization patterns in every of your aspects. Yeah. We have another question about volumetric printing. So they want to know, so volumetric printing seems to combine the features of tissue culture in bioreactors with monolayer culture / additive printing. Is that a correct assumption?
I'm not sure I understood exactly what the person asked the question mean, but if I understood correctly, I wouldn't really say that it combines bioreactor culture and monolayer culture. You can create objects like a porous object that you can then place in bioreactor, for example. So you have a step before the bioreactor itself. When the cell suspension that then becomes the object after photoplastinking is placed into the vial of the printer, that's no reactor, because you don't have any mechanical stimulation or no perfusion in there. You just have the illumination coming from the different angles. But then that same object can indeed then be taken and placed, for example, in a perfusion chamber, and then you can have the bioreactor function. But that's more of a post-printing effect. It's not it's not in the print itself.
Yeah. So this question is coming to both of you again. How common is the use of bioprinting in the field of disease modeling? I think this is something even I'm very interested to know, especially in tumor organoid models. So, Dr. Javier, do you want to start?
Yeah. I mean, it's a really starting field. So again, that's there is a lot of promise there, because also this is also related with the ethics behind, right? There is a growing call for kind of reduced animal modeling, right? So and organ printing and disease modeling, right, kind of like is moving into that direction. So and also not only for the ethical issue, also for practical issues. There is many technologies and many therapies that they do amazingly in animal models, but they fail when they move to the humans, right? So the animal models, they start to get like some kind of ethical concerns, and also they are not that good. So the reproduction of organ models for disease modeling, right, is actually like being pushed towards like these very strong forces, right, to do that. Is not there. I mean, that is, I know, for example, now with the coronavirus, there was like many people using organoids to actually like test the effects in different parts of the body, and again, in controlled environments and things like that, right? And it's been like more and more popular. It's definitely like a really hot topic, but is still being like a lot of questions about like how reliable is this, right? It's very promising. We are seeing the reproduction of very specific functions and very rare functions of organs in the models, right? But again, that's is a really young field actually.
So I do want to bring this the next question that I will be asking pertaining to the ethical aspects. But Dr. Ricardo, do you want to add on to what Dr. Javier just said?
Yeah, very briefly. Indeed, it's like this. So it's a very young field, and the main question that remains is the predictive power, how strong that is, especially because when you have an animal model, it's not a good model of a human, but you have the whole organism, of course, that you can look at. What is happening though is that at least in the US FDA and Europe, the EMA, they are starting to consider the use of, for certain aspects, the use of organ on chip, for example, technologies. And there is legislation will start to build around that, but I think that there will be a gradual step. So the first thing that will happen will be a complement to animal experimentation, mainly to rule out those drugs that shouldn't be tested in human, and without the need to test them in animals as well, for example. So it's sort of an approach like fail fast and fail often, so that you don't have too many candidate drugs that go to the clinical trials and then they don't work. So you just get rid earlier of the non-promising drugs, let's say. That's probably the first thing that's going to happen. And then as this model improve their predictive power, or actually that's actually confirmed, then there will be likely a gradual replacement, entire replacement of animal experimentation.
So that brings me to an intriguing question. So as I was thinking, so you mentioned about the ethical aspects. So what are the regulatory challenges that you are facing or will face, or how is the regulatory landscape when it comes to bioprinting?
I can start. So I mean, I think all the regulatory aspects, they are common to the whole tissue engineering, right? I mean, in a sense, all that the things that we are doing with bioprinting are basically like a branch of tissue engineering. And I mean, the most challenging, or like the area that seems to be more controversial, is about like the uses of stem cells and the origin of those stem cells. That's not only like a part of bioprinting, it's a really like a general issue in many areas of medicine, right? As I say, I think that there is also, I mean, obviously there are some controversial parts. Also, we need to be like to see also the positive side of it. There is many ethical issues that bioprinting might solve, right? So, for example, the test of drugs in humans, or the test of drugs in animals, or like the simplification of all those processes, right? So I think that there is, in this case, what I see is that they are, the bioprinting might actually like help solving some ethical questions in other places rather than bring new ones to the table. But that's in my opinion, and again, like we are also talking about like in the how we envision bioprinting is that we will be using the same cells from the host, right? That is one of the amazing things that is not possible with current organ transplant. That is the idea that you can take cells from a person, like going back to the stem cell states, and actually start producing these organs with the genetic of the host, right? And that actually like remove all the idea of like especially like certain religions that they don't allow to have organs from other people, or people from actually like other religions, right? So that's actually what, from my perspective, is solving, or is going to solve more issues than the ones that is going to open.
So in brief, it's not yet tightly regulated, right, from what we understand. It is not, yeah. Okay.
That's correct. I also, from what we see, that there is no actual specific regulation for bioprinting. There exists regulation for tissue engineering. So that's, at least in Europe, it falls under the advanced therapeutic medicinal products, which have a particularly strict, like most cell therapies, they have a particularly strict regulatory approval. But there is no extra constraint for the fact that there is printing involved. There is one thing I maybe would like to underline on the ethics side, is that it's true. I also believe that it solves many more issues that it can bring. But it's important that there is continuously good communication with society, because of course some people may not see that as a feasible solution yet, whereas it goes in that direction. From our university, in collaboration with the actually bioethics department, some years ago, we ran a series of questionnaires in the Netherlands regarding the feeling about bioprinted ears. So basically, a ear prosthesis that it's mostly aesthetic, so it's not a life-threatening conditions. Not having an ear, of course, it has a lot of problems, but it's not life-threatening per se. And the vast majority of the people that were interviewed are, of course, extremely excited about the technology and this perspective. But only a minority of them was actually willing to say, okay, if I needed, I would want a bioprinted ear to be implanted in me. So that there is still, I think, a bit of a cultural gap that needs to be filled through information mostly, and through open communication between academia, industry, and the general society.
So how far do you think we're away from commercialization, actually? Maybe that'll be an appropriate question to ask based on the legal and regulatory areas that you just talked about.
If we talk about in the pharmaceutical industry, so in vitro models for the drug discovery route, I don't think we are that far. There is still work to do, but some major companies are already investing or expressing interest in that. So that's not that far. The medical transplantation field, that's a bit further away. But in general, because we are still missing some fundamental biological development that can help us get to those complexity that we observe in vivo. So it's way different to make an organ for transplant rather than making an organ on chip, for example.
Absolutely. So, Dr. Javier, do you want to add anything to this?
No, I think that Ricardo answered the question in a very clear manner. I think that also something that is important to remark is that this is not going to be like suddenly we are going to see like all 3D printing. I mean, definitely this is going to be a gradual process. We are going to start seeing like different organs for different applications, right? So I think that one of the fields that is going to benefit like immediately is definitely like bone. Bone has been there for a long time, and is something that it really need to be specific to the patient and specific to the repair that you want to do, right? And but again, it's not going to be that suddenly all the organs are going to be printed, right? Or all the chips are going to be made. So we are going to see like how we succeed for very specific organs, very specific disease, or very specific applications, and it's going to be becoming like more and more part of our life, right? So it's not going to be a shock in any case, or I don't foresee it being a shock.
I agree completely. So following my question, I think there's another question from the audience. What do you think would be the most feasible application of bioprinting in the next 10 to 20 years to come? Anyone can pick it up.
I think I just mentioned that. I think that for me, bone is going to be one of the first ones. If you remove like everything that is related to prosthetics and things like that, that is already there, right? So it's a little bit already entering into the field, right? I think that bone is, I mean, it's a little bit more, how to say, is a little bit more open to tune, to get different materials, to basically let it develop, right, in a sense, right? So to focus a little bit more into the mechanics and then let the whole process to a little bit be embedded. So I think that if I need to put my money somewhere, that is where I will put it. But again.
I'd agree on the, definitely bone is one. I think that there's 3 candidates in my view that would be bone, cartilage, and skin. That would be my, if I can bet on 3 horses, that would be one of those in my view. Again, for the matter of it did relative simplicity compared to other organs like heart or like liver, for example.
So we have at least 3 or 4 that are coming in the pipeline, I guess. So the next question is, again from the audience. We could see bioprinting is advancing with multiple advancements. Here, what is the status of biomaterials? Is there a particular hydrogel that can mimic all organs? Particularly interested to know what is the research gap in terms of biomaterial development?
Yeah, that's a very important point because on one hand, I think that the technologies and engineering side from the device or the printing technology has been the field that moved the faster up to now. The biomaterial side still needs a bit more of a tuning in that sense. I think that I don't think there is such a thing as a universal ink or universal hydrogel that will fit all the cells. At least there is no such a thing now. Maybe there will be in the coming future. I believe that synthetic or pseudo-synthetic materials, so based on ECM matrix like gelatin-based or other laminin-based or whatever else protein-based materials will be the most important in this field. But there is of course the possibility to add growth factors and other things that needs to be considered. But there is definitely a need, because right now the best materials for cell culture in general are still matrix mixtures for like matrigel that are derived from mouse tumors or something like that, that are not suitable for many human-based applications. So there is a need. There's been a huge leap also in materials development, but there is still a lot to do, I think.
Right. I think, I'm not sure if this is the last question, but in the interest of time, I think maybe we can do two more questions. I think this question is directed to both of you. What is the maximum dimension that you have been successfully, you have printed so far? What's the maximum? It's more technical, so they would like to know the dimensions.
That's actually like a really good question, because one, and again, is related to the vasculature, right? In my case, the biggest thing that I have produced is like centimeter scale, right? But that's a trick, because the thing is, like, your thickness is the one that is limited, like what you can do. I, for example, can make a tube, right? Or like a vein mimic. But the problem is that, and I can make it like 1 centimeter long. But the problem there is that if my walls are too thick, then my cells, they are not going to get enough nutrients, right? So, and then they are going to start dying. So the definition of how big depends a little bit of what you, where do you define the dimension? So, and again, the problem is always going to be like the movement of nutrients and the movement of oxygen, right? So, so it's very rare to see anything that goes beyond the half millimeter to the millimeter in terms of thickness. So you start to get like serious problems there.
Penetration issues. Mm-hmm.
Yeah, indeed, we are also in that range. I think a bit above a centimeter cube in volume, but they are all porous constructs. So you need to look. If you look into them, it's a combination of pores with very thin walls, basically, that indeed are below the millimeter when it comes to the individual components. And all assembled together, it's bigger than a centimeter cube.
Mm-hmm.
I think that also is something that maybe and the audience didn't get, right, and then and it's really important and related also with the material question that we had before. But when we print an organ, we don't print like the final organ, right? There is a time for like the cells. When we print, we need like something that is like a fluid, and cells, when they are in an organ, they are like perfectly attached to each other and they're interconnected, and they are actually like a solid, right? So what we do is that we put the cells where they need to be, right? But there is a time after that actually like the organs builds on its own, right? So we need this property of life, of actually like grow and develop, right? So and that's the thing. So it's like if you do this, even if you print like something really big, it's going to be porous, it's going to be spaces, right? And the amount of cells that you are putting is definitely not the amount of cells that you expect to have at the end, right? So there is this process between the printing and your final result that is based on the development of the organ, right?
Thank you. Maybe we'll just do one last final question for today. So for materials manufacturing techniques, we typically rely on ASTM standards. 3D bioprinting, on the other hand, is progressing at a faster rate. I understand that maintaining cell viability is an important bioprinting criterion. However, what standards and benchmarks can be used to test bioprinted constructs for cytotoxicity and define material printability?
Yeah, that's also a very relevant question. I don't think there is an ASTM standard for bioprinted device. There are ASTM for 3D printed objects. For bioprinted device, I would still, if you want to follow the ASTM, I would still follow the cytotoxicity development for testing of material that exists in the STM standards. That being said, for now there is no specific recommendations when it comes to bioprinted constructs. So it's a bit up to which type of testing normally do in your lab for that. So there is no real guidelines yet that come from regulatory bodies.
Yeah. Also, I mean, there's obviously regulation depends on the application that you are going to use, right? So it's not like your regulation or your test, they are not the same if you are printing a heart or you are printing a bone, right? So yeah, but I think that even for materials, we are still using STM for like all the measurements. Like everything that is related to bioprinting is related to viscosities and rheology, right? And we are still using the same ASTMs that people use for biological materials. I mean, there are like tons of STMs for glues, for materials for implants, for all of that is there, and we just borrow them from those that already exist.
So thank you so much, Dr. Javier and Dr. Ricardo. I think we could easily go on for another half an hour, but in the interest of time, I will have to stop here and pass the platform back to Jin.
Thank you.
Thank you. Thank you so much, Anya, and to Ricardo and Javier for the great sharing. And thank you to everyone in our audience for your ongoing questions. It is always very heartening to see so much enthusiasm at our online sessions, but real conversations often happen after, so we do hope that all of you can keep connected. The recording of this session will also be uploaded on SGInnovate's YouTube channel, so do head on there for a review of today's event and till our next online event. Thank you and bye, everyone.
Thank you.